Imagine for a second that you are looking at a map of a city, like a standard old-school fold-out map.
Yeah, the kind you'd keep shoved in a glove box.
Exactly. You can see the broad neighborhoods, right? The major highways, maybe the big parks. But now imagine upgrading that map to a satellite view so incredibly powerful that you can zoom in and see individual alleyways.
Like street-level definition.
Right. You can literally see the structural cracks in the foundations of the houses. You can even see which streets are hopelessly congested and which have been, completely abandoned.
It's a totally different way of seeing the same space.
It is. And that leap in resolution, that sudden breathtaking clarity is exactly what we are exploring today on this deep dive. Only the city we are looking at is the brain.
Specifically, a brain in the early stages of Alzheimer's disease.
Right. We are diving into a really groundbreaking February 2026 paper. It's from Nature Neuroscience, titled Magnetic Resonance Microscopy Maps Widespread Effects of Alzheimer's Disease on Brain Structures and Behavior in Mice.
Quite a mouthful, I know.
It is, but the mission of our deep dive today is to unpack this massive microscopic atlas, literally region by region.
Yeah, we want to figure out exactly what happens to the brain's real estate and, well, why everything we thought we knew about brain shrinkage might actually be hiding a much stranger truth.
Right, because it really is a complete paradigm shift.
Right.
But before we even look at the new map, I think we have to talk about who we are actually mapping.
Oh, absolutely. The subjects matter just as much as the technology.
Yeah, because when I was reading through the background notes, it struck me that historically, Alzheimer's research has had a bit of a severe diversity problem when it comes to the test subjects.
You're talking about the mice.
Yeah. We test potential cures on mice, but those mice haven't always been the best stand-ins for humans. We've basically been using genetic clones, right?
That is a crucial place to start.
Yeah.
I mean, for decades, preclinical trials, meaning the tests we run on animal models before a drug ever touches a human being, have relied heavily on a single, highly inbred genetic background.
Like testing a miracle drug on an army of identical twins.
Exactly. The classic example in neuroscience is the C57BL6J mouse. It's generation after generation of inbreeding until every mouse in the lab is practically identical, which, is incredibly efficient for controlling your variables in a lab.
But terrible for translating those results to human beings, because humans are definitely not clones.
Right. If you test an Alzheimer's drug on a single genetic profile, you might find a compound that perfectly cures that specific profile. But then you run a clinical trial on a genetically diverse human population, and the drug just fails completely.
Because the underlying genetics of the patients are reacting differently to the drug.
Exactly. We actually see the profound impact of genetic diversity in human Alzheimer's data all the time. Take the APOE epsilon 4 gene variant.
Okay, yeah, I've heard of that one.
It's widely known as a massive genetic risk factor for Alzheimer's. But the risk is not a universal constant.
Wait, how so? If you have the gene, don't you just have the risk?
Not necessarily.
Yeah.
So in people with Asian ancestries, inheriting that epsilon 4 variant carries incredibly strong deleterious cognitive effects. I mean, it is a major red flag.
Okay.
But if you look at populations with several African and Hispanic ancestries, someone could inherit 2 copies of that exact same gene variant, and their Alzheimer's risk, well, it barely registers above the background average.
Wow. Are you serious?
Yeah, the background genetics, like the thousands of other genes interacting with that one mutation, they completely change how the disease manifests.
So the disease is like a match, but the genetic background determines if you are dropping that match into a puddle of water or like a pool of gasoline.
That is a perfect way to visualize it. So the researchers in this 2026 paper, they knew they couldn't use the clone army anymore. They had to build a better blueprint.
Right, they needed diversity.
Yeah, so they used a population called ADBXD F1 hybrids.
Okay, let's unpack the mechanics of that, because how do you intentionally build a genetically diverse mouse model of Alzheimer's?
Well, you start with a mother who carries what is called the 5xFAD transgene.
5SFAD, okay.
Right. This means she was genetically engineered to carry 5 highly penetrant human mutations that are known to cause severe early-onset familial Alzheimer's disease.
And what are those mutations doing?
So 3 of these mutations are in the APP gene, which stands for amyloid precursor protein, and 2 are in the PSEN1 gene, or presenilin-1.
Okay, wait. What do those specific genes actually do? Like, why those 5?
Think of APP as a normal protein sitting in the cell membrane, Usually gets cut up by these little molecular scissors and cleared away without an issue.
Just normal cellular housekeeping.
Exactly. Now, PSEN1 is a critical part of those molecular scissors.
Yeah.
When you have these 5 mutations, the scissors malfunction, they cut the APP protein in the wrong place.
Oh, and that creates the plaques.
Right. It produces these highly toxic, sticky amyloid beta fragments that rapidly clump together. These mutations basically act as a fast-forward button for amyloid plaque buildup.
Which I guess you need, because a mouse doesn't live for 80 years to develop it naturally.
Precisely. We have to fast-forward it. So the mother brings the severe, fast-forward human Alzheimer's mutations.
Yeah.
But to get the diversity, she needs to be crossed with different fathers.
Right. So who were the fathers?
They crossed her with males from 4 distinctly different genetic strains, known as BXD strains. Specifically, BXD 32, 65, 77, and a 101.
Just a whole mix of genetic backgrounds.
Yeah. And the resulting offspring, that first generation, they all inherit the severe Alzheimer's mutations from their mother, but they have 4 entirely different genetic backgrounds from their fathers.
That is brilliant. They successfully built a diverse population that actually mimics the messy reality of human genetics.
Exactly.
Okay, so we finally have a realistic population of mice. But this brings up a logistical issue that honestly blew my mind when reading the source material. A mouse brain is tiny.
Very tiny, roughly the size of a pea.
Yeah. How on earth do you map a pea-sized organ in enough detail to see the structural cracks in the alleyways, to use our earlier analogy?
You have to push imaging technology to its absolute physical limits. They use something called magnetic resonance histology, or MRH.
How is that different from a normal MRI?
Well, you've probably heard of an MRI at a hospital. Those machines typically use a 1.5 or maybe a 3 Tesla magnet. These researchers used a massive 9.4 Tesla vertical bore magnet.
Whoa, that is an absolute beast of a machine. The magnetic field must be incredibly intense.
It is so powerful that it allows them to use a technique called diffusion tensor imaging, or DTI, which is mind-boggling precision. They took 61 angular samples.
What does that mean, angular samples?
Basically, they scanned the brain from 61 different directions. And the magnet is so strong it can track the microscopic diffusion of water molecules as they bounce off the walls of cells and nerve fibers.
So they are tracking the water inside the brain tissue.
Yeah. By tracking how the water moves, they can draw the exact 3-dimensional shape of the tissue itself.
And what kind of resolution does that actually give you?
25 micrometers.
25 micrometers. Just to put that in perspective for anyone listening, a standard human hair is about 70 micrometers thick. They are capturing 3D pixels or voxels that are roughly a third of the width of a human hair.
It's unbelievable.
Wow.
And because of that granular resolution, they were able to perfectly delineate 231 distinct regions of interest across the brain.
231 regions in a pea-sized brain.
Yeah. They could measure structures as tiny as 0.1 cubic millimeters, like the trapezoid body in the brainstem. The precision is so surgical that when they compared the measurements of the left hemisphere to the right hemisphere, the error margin was a microscopic 1.8%.
That is just wild. Okay, let me just pause and make sure I'm holding all of this in my head, because the sheer scale of this is a lot.
Go for it.
So, bullet 1. We've moved away from testing on genetic clones and created a diverse hybrid mouse that actually reflects human variability. And bullet 2. We threw those brains into a 9.4 Tesla magnet, mapping water molecules to create a 3D atlas with 231 distinct regions, all at a resolution smaller than a human hair.
You nailed it. That is the foundation of the study.
And I want to throw a question to you, the listener, really quick. Think about this. If your genetics fundamentally dictate how a disease manifests, how much of traditional single-strain medical research might we have to rethink entirely?
It's a big question. It makes you reconsider the last few decades of medicine.
It really does, which makes the actual findings of this map so crucial. So we have this ultra-high definition view. The mice are 14 months old, which in this fast-forward model means they're deep into the disease progression.
Right, they are severely affected at this point.
So when you finally put the healthy control brains on a scale against these severe Alzheimer's brains, what does the disease actually look like?
Well, based on everything we traditionally know about Alzheimer's, you would expect the diseased brains to be significantly smaller, right? Because the defining characteristic of the disease is brain atrophy, shrinkage.
Right, you always see those comparison images in textbooks where the Alzheimer's brain looks withered.
Exactly. Well, here's what they found. The total volume of the healthy control brains was 477.8 cubic millimeters. The total volume of the Alzheimer's brains was 475.7 cubic millimeters.
Wait, a difference of 2 cubic millimeters?
Yep.
That's barely 0.5%. That is statistically nothing.
It is nothing. The total brain volume is virtually unaltered. There is no global overarching effect of the 5xFAD mutations on the overall size of the brain at this 14-month mark.
I really have to push back on this because it feels counterintuitive to literally everything we're taught. If the brain isn't shrinking, how is it Alzheimer's? Where is the damage?
Oh, the damage is profound, but it's hidden inside the skull.
Huh.
See, the total volume doesn't change, but over half of those 231 individual brain regions shifted radically in size.
So they are changing internally.
Right. It is a zero-sum game. The brain is literally reorganizing its internal real estate along what we call a rostrocaudal gradient.
Okay, define rostrocaudal for us. What exactly is happening?
Rostrocaudal simply means front to back. So the front of the brain contains the telencephalic regions. This includes the neocortex, the hippocampus, the amygdala, and the olfactory cortex.
Basically, the centers for higher thinking, memory, and emotion.
Exactly. And in these Alzheimer's mice, those regions don't shrink. They unexpectedly swell. Some of these memory centers gain up to 10% in physical volume.
They get bigger. That makes no sense.
It's a total paradox, right?
Yeah.
But remember, the skull is a fixed, rigid box. It doesn't expand.
Right.
So if the total volume of the brain isn't changing, but the front of the brain is ballooning by 10%, that swelling has to be counterbalanced physically somewhere else.
Nothing has to give.
Exactly. So the deeper back regions of the brain, the thalamus, the brainstem, the hindbrain, and the major white matter tracts that route the signals, they shrink significantly. They're essentially crushed to make room.
I am still completely stuck on the memory center swelling. I mean, why would the cortex and the hippocampus, the exact places where Alzheimer's does the most cognitive damage, be expanding?
We have to understand the concept of prodromal, or early stage disease. This mouse model mimics the early stages of human familial Alzheimer's. And surprisingly, human neuroimaging data back this up.
Really? Humans do this too?
Yeah. When researchers look at human patients who carry the PSEN1 mutation, before they even start showing memory loss, their cortex is actually 250 micrometers thicker than a healthy person's.
That's fascinating.
It swells early on before it eventually crashes, burns, and atrophies in the late stages.
Ah, okay. So it's a phantom swelling. It looks bigger on the MRI, but it's not actually growing new, healthy neurons.
Far from it. The swelling is an illusion, hiding catastrophic cellular death.
How do we know that for sure?
To prove this, the researchers didn't just rely on the MRI. They used a secondary technique called light sheet microscopy, or LSM. And this is where the science gets almost like science fiction.
Oh, I love this part.
They literally use chemical solvents to strip the opaque lipid fats out of the brain tissue, turning it completely transparent like a piece of glass.
They make the brain invisible.
Essentially, yes. And then they use a specific fluorescent tag called NuN.
NuN.
Right. And NuN binds only to mature, healthy neurons. So they shine a microscopic sheet of laser light through this transparent brain to physically count the glowing cells.
So they bypass the swelling entirely. They can look straight through the inflamed tissue and count the actual survivors.
Exactly. They zoomed in on the CA3 region of the hippocampus. Now, on the high-def MRI, the CA3 region had clearly swollen, right? It gained volume.
Right, it looked bigger.
But the light sheet microscopy revealed a 25% loss of NuN-positive pyramidal neurons. A quarter of the mature memory cells in that region were entirely dead and gone.
Wow. So what is making it bigger if the cells are dead?
Inflammation. When those neurons start dying from the toxic amyloid plaques, the brain's immune system panics. Cells called glia microglia and astrocytes rush into the area to treat and clean up the damage.
Like an emergency response team.
Exactly.
Yeah.
You get massive structural inflammation and fluid buildup. The glia are physically taking up more space than the neurons they're replacing.
You know, the way you're describing this, imagine a house where the total square footage stays exactly the same.
Okay.
But suddenly the living room starts expanding. It gets 10% bigger. But it's not magic, right? It's expanding by crushing the hallways, the plumbing, and the electrical wiring in the walls just to make space.
I like that.
And when you finally look closely at that bigger living room, you realize the beautiful hardwood floors are actually rotting away, and they're just covered up by this cheap, swollen, inflamed carpet.
That is a brutally accurate analogy. The living room looks larger to a casual observer, but the structural integrity is failing from the inside out.
Right. Let me do a quick recap here to synthesize this second massive concept before we move on.
Go ahead.
So, bullet 1, total brain volume remaining unchanged is a dangerous illusion. Bullet 2, the disease operates as a zero-sum reorganization where the front of the brain swells with inflammation, crushing the regions in the back. And bullet 3, That swelling is actively masking the mass death of actual memory cells.
Yes. And if you don't mind me jumping in, that is a terrifying thought when applied to human medicine.
How so?
Well, if a neurologist is looking at a standard hospital MRI and they see a brain that hasn't lost any overall volume, they might think the patient is perfectly healthy.
Oh, wow. They wouldn't see the zero-sum shift.
Right. They could easily miss the fact that the internal architecture is actively collapsing under the guise of inflammation.
Which brings up a great question for you, listening to this. Imagine living in that house where the square footage stays the same, but the walls are crushing the plumbing. How long until the house simply stops functioning?
Not long at all.
And that structural collapse brings us to the next critical piece of the puzzle. We've talked about the rooms of the brain shifting size, the swelling, and the crushing. Let's talk about those hallways I mentioned in the house analogy.
The electrical wiring.
Exactly. What happens to the communication pathways connecting these shifting regions?
The white matter, the massive bundles of nerve cables that send signals across the brain, takes a devastating hit.
Because they're being crushed.
Partly, yeah. The researchers focused heavily on the corticofugal projections. These are essentially the main communication highways, sending commands from the thinking cortex down to the rest of the body.
Okay.
The corticospinal tract, for example, lost up to 8.1% of its volume.
So the physical wires are getting thinner because they are being squeezed by the swelling above them.
They are getting thinner, yes, but the physical shrinkage isn't even the worst part. The quality of the wiring degrades.
What do you mean by quality?
To measure this, they used the diffusion MRI we talked about earlier to calculate something called fractional anisotropy, or FA.
Okay, let's break down the mechanism there. What exactly does fractional anisotropy measure?
It measures how smoothly water flows along a nerve fiber. Healthy nerves have a tight protective coating of fat and protein called myelin.
Oh, right, like the thick rubber insulation on a copper electrical wire?
Perfect comparison. When that myelin insulation is healthy, water flows strictly in a straight line right down the pipe. We call that high fractional anisotropy.
Because the water is completely contained.
Precisely. But when the myelin is damaged, when it degrades or frays, the water isn't contained anymore. It leaks out radially to the sides.
It bleeds out.
Yeah, we call that low FA. In these Alzheimer's mice, the FA in those major descending tracts dropped by an enormous 11%.
So the cables aren't just being crushed, they are literally fraying. The insulation is rotting away and the signals are leaking out.
Exactly. And this is the moment where we have to put those 4 different fathers back into the picture.
Right, the genetics, the BXD sires.
Yeah, because this damage we're talking about, it wasn't uniform across the board.
How much did the genetic background noise actually dictate the severity of the fraying and the shifting?
Immensely. The genetic background of the mice explained a full 23% of the variance in the volume changes across the brain.
Almost a quarter of the difference is just who your dad is.
Exactly. Let me give you the starkest contrast from the data. The offspring that inherited the genetic background from the BXD77 father were hit incredibly hard.
What happened to them?
They exhibited significant volume changes in 124 out of the 137 affected brain regions.
124 regions shifting. That is almost a total architectural collapse of the entire map.
It is. But look at the offspring from the BXD65 father. They only showed significant structural changes in 26 regions.
Wait, they all had the exact same severe fast-forward Alzheimer's mutations from the mother, right?
The exact same mutations. But the severity was drastically mitigated based entirely on the background genetics of the father.
That is wild.
The study also noted another massive variable: sex. Female mice consistently showed much higher variability in their regional volume changes than the males.
So it's not a one-size-fits-all disease.
Not at all. It underscores that Alzheimer's is not a monolithic disease. Sex and genetics completely alter the map's destruction.
Okay, so we have a brain with swelling rooms, fraying wires, and a severity dictated by a genetic lottery. But what does this microscopic shifting map actually mean for the living, breathing mouse?
You mean their actual behavior?
Yeah. Does all the structural reorganization translate to how they act?
It does, and the researchers mapped the structure to the behavior perfectly. They ran the mice through behavioral tests called contextual fear acquisition, or CFA, and contextual fear memory, or CFM.
CFA and CFM. I'm assuming that involves teaching the mouse to be afraid of something.
Yes.
How do you actually test a mouse's fear memory?
You place the mouse in a very distinct chamber, maybe it has a specific smell or a unique grating on the floor, and you give it a mild foot shock.
Ouch. Okay.
A healthy mouse learns instantly. It acquires the fear of that specific context. That's contextual fear acquisition. CFA.
Right.
Then, when you put the mouse back in that exact same chamber 24 hours later, it remembers the shock and it freezes in place. That's contextual fear memory, CFM.
And how did our Alzheimer's mice with the reorganized brains perform?
The researchers found exact, highly specific correlations between the shifting regions and the cognitive failures.
Can you give an example?
Sure. For instance, there is a major white matter fiber tract called the fimbria. They found that mice with a physically smaller, shrunken fimbria directly correlated with worse fear acquisition.
Because the wiring was frayed and crushed.
Exactly. The mouse literally couldn't process and learn the context of the fear in the first place.
And what about the memory test 24 hours later?
For memory, they looked at a specific subregion called the hilus of the dentate gyrus. In the Alzheimer's mice, this region was actually larger.
Oh, part of that phantom inflammatory swelling we talked about.
You got it. And the data showed that the larger that specific region swelled, the worse the mouse performed on the fear memory test the next day.
Wow.
The physical microscopic changes mapped one-to-one with the cognitive decline.
So the structural collapse absolutely predicts the behavioral collapse. Let me do one final synthesis of the mechanics here.
Go for it.
Okay, bullet 1. The white matter cables lose their myelin insulation, causing the brain's signals to leak out. Bullet 2. The severity of this damage is completely at the mercy of your background genetics. One lineage saw 124 regions fail, while another only saw 26.
Spot on.
And bullet 3. Every millimeter of that shifting, swelling, and fraying directly robs the animal of its ability to learn and remember.
You've captured the entire mechanism perfectly. Again, this circles right back to you listening to this.
Think about your own deeply unique genetic background. If a revolutionary new Alzheimer's treatment is developed, but it's only ever tested on a clone army or a single genetic profile, what are the odds it works flawlessly for your specific complex biology?
The odds are incredibly slim, and that really drives the urgency of this research home.
It really does. So as we zoom out and put this entire map back together, what is the ultimate value of this 25-micron ultra-high-definition atlas we've spent the day exploring?
The value is that it fundamentally rewrites the textbook on preclinical research. We now have undeniable proof that early-stage Alzheimer's is not a simple linear story of a brain slowly shrinking away into nothing.
Right.
It is a highly complex, genetically dependent, zero-sum reorganization. It is a terrifying dynamic where toxic inflammation causes the front of the brain to swell, hiding the mass death of memory cells, all while crushing the vital pathways in the back of the skull.
And the reason we should care about the precise volume of a microscopic pathway in a 14-month-old mouse is because establishing these benchmarks is the foundation for finding a real cure.
Exactly.
When we run drug trials now, we don't have to guess if a drug is working based on a blurry scan or, a subjective behavioral test. We have the high-definition map.
We can administer a new therapeutic, put the mouse to the 9.4 Tesla magnet, and see exactly down to the micrometer if that drug stopped the living room from expanding and if it stopped the wiring from fraying.
It provides the ultimate objective diagnostic tool for the next generation of therapeutics.
We can finally see the disease clearly.
We can finally see the cracks in the foundation. And honestly, looking at this map leaves me with one final, deeply provocative thought.
What's that?
We learned today that the physical total volume of the brain remained completely unaltered. The container itself didn't change size.
Right.
Yet the internal geography shifted so dramatically that it completely erased the animal's ability to learn and remember its own environment. It begs a profound, almost philosophical question.
It really does.
Yeah.
Because it makes you wonder what a memory actually is.
Right. Is the essence of a memory stored physically inside the cells themselves? Or is a memory merely a fragile product of the precise physical distances, the tension, and the structural architecture between the different regions of the brain?
Wow.
When the map shifts, does the memory die because the house collapsed, or simply because the hallways no longer lead to the right rooms?
That is a brilliant way to frame it, and something we will be investigating for a long, long time.
It is definitely something to mull over. Until next time, keep zooming in on the map.